Hello everyone, I'm presenting this work, Development of Bioinformatics Platform for Microbiome Metagenome Analysis. So I'm going to start a little bit with the introduction. Metagenomics is an independent culture method that allows the study of different microorganisms from an environmental sample. DNA is extracted, sequenced, and the results are analyzed using computational tools. This approach represents the best option to access the microbial genetic diversity. Bioinformatic analysis of a metagenome sequence is divided into phases, assembly and binning. Assembly refers to the construction of consensus sequence denominated contigs from the short reads generated in the sequence. To obtain a correct assembly, there must be plenty of overlap between the reads. On the other hand, the binning phase is a process where contigs are assigned to taxonomic groups. Next-generation sequence has allowed the exponential growth of data from metagenome studies. Therefore, it has been necessary to improve the computational methods for this data interpretation. Despite the fact that multiple software have been developed with this purpose of reconstructing the original metagenome, it is still necessary to focus on comparative studies, benchmarking, to determine software capability for the reconstruction of the original scenario. We search for studies that perform a molecular characterization of microbiota of soils contaminated with heavy metals. We select 16 species of bacteria. With this initial dataset, 10 synthetic metagenomes were prepared on Illumina type data using the NEST program, which allows the generation of NGS data. In this context, 5 synthetic metagenomes were developed based on the number of genomes and their phylogenetic distance. In addition to this, for each synthetic metagenome, 2 simulations were performed, varying the abundance of the species. For each metagenome, there are 2 types of abundance, scattered and balanced. We select 8 software for this assessment, which were selected based on their characteristics, objectives, and status. The 4 programs for the assembly phase are MegaHit, BrainMeta, Spades, and Velvet, and for the binning phase are ConcuCT, MetaBat, MaxBin, and MyCC. The development of the bioinformatic platform allowed it to automate the simulation, assembly, and binning process. The platform was designed using Python, and the main characteristic is that the 8 software can work jointly, allowing the contrast of the results. So we also consider different metrics for each of the phase. And for the assembly stage result, I'm going to talk about this metric that is a genome fraction, which refers to the total number of aligned bases in the reference divided by the genome size. We can visualize through this graph and this graph that Spades and MegaHit maintain a similar behavior. Among the reasons why these efficient results can be attributed is that both programs are based on algorithms that include strategies like the use of different sizes of cameras in the case of Spades, and also the multiplier and solid immersive camera for the case of MegaHit. However, the evaluation of the other metrics shows that Spades maintains high values, for instance, on the interspecific translocations. Therefore, it is inferred that MegaHit managed to overcome Spades. We also obtained that these results were also assessed by this study. Therefore, we conclude that MegaHit has better sensibility, and it can lead to a genomic reconstruction of better than 50%. For the binning phase, we also consider different combination of the programs, and the software that obtain the best results correspond to the combination of MegaHit and MyCC in at least 3 categories. Furthermore, it is indicated that for the reconstruction of metagenome is superior as the diversity and the number of samples increase. The results generated in this research can indicate that MyCC gained an advantage over other programs. MyCC is characterized by accurate binning because it is configured in such a way that works even with a small sample size, from 1 to 11 genomes. Therefore, simulated metagenome samples being an average of 8 to 16 genomes have offered a certain advantage for the performance of this software.